have the great pleasure of introducing Carly Kenkel. Dr. Kenkel is a research professor at the University of Southern California and got her PhD at University of Texas, Austin. She studies the genetics of coral adaptation, diversity, and response to climate change. She's the 2019 Sloan Research Fellow, 2020 ICRS Early Career Award. She's a key member of our CRC Genetics Working Group. She's written about 80 papers, which is pretty incredible for a young professor. 8 of them were written in 2021 as she was just twiddling her thumbs with 2 small children during COVID. And my favorite paper during that period of time was supporting mothers during COVID and beyond. So she has definitely written about things outside of just coral genetics. And my favorite accolade is on Rate My Professor, she has the grade of awesome with the quote, "She only taught half the class. I wish she taught the whole class." So please welcome Dr. Kenkel to the stage.
Thank you, Dolly. That's some hardcore stalking. I appreciate that. It's my great privilege to be here today, and as a member of the Genetics Working Group, I've actually been tasked with sharing with you our latest product, which is a perspective piece on managing expectations for the selective breeding of future corals, or as we've come to think of it amongst ourselves, the supercoral myth. And I just want to preface this by saying I am a geneticist, and as Tali pointed out, I am a professor, so there's going to be some learning involved today. Someone said to me, "Wow, you're really getting into the weeds there." And I said, "That's okay. This is an intelligent audience. They can handle this." So buckle up, folks. And also, before I go any further, I really need to acknowledge the efforts of my brilliant colleagues. I'm sad to say not all of whom can be here today because they're actively dealing with the hurricane. It's my very great pleasure to be able to talk with them, think with them, and what I'll be telling you about today represents very much a consensus of thought only achieved after many hours of intense discussions. So when I refer to the myth of the supercoral, that's not to dismiss the idea as some sort of fabrication. We have many examples of individuals, populations, species, even reefs that appear to defy the odds, like this fully pigmented coral in a sea of bleached conspecifics. And we want to be able to take that superness, incorporate it into our restoration programs, and ideally improve upon it, right? We want to be able to select the most temperature-tolerant subset of our nursery population, breed them together, and in the next generation see an improvement, an increase in the trait value in the offspring. And this isn't a wild idea. Selective breeding has been hugely powerful, particularly in agriculture and animal husbandry. And so I want to share with you an example that I really love. It's actually the longest running selective breeding experiment from a crop plant. This was an experiment started at the University of Illinois in 1896, and what those researchers were trying to do was increase the oil content of their corn in their plot of corn. So this is showing you the data that they started with in 1896. Y-axis is number of individuals, x-axis is percent oil content for that particular plant. And so it's showing you that on average, back in 1896, oil content was about 5%. And what they did, they selected the plants with the highest oil content to use as seed stock for their next generation. If you could just start the animation. What you're going to see now is how that oil content has changed over time. And what I really find fascinating about this is not only is oil content increasing and increasing quickly, but look at how far they managed to push the trait value beyond just the original range of variation, right? It started at 5%. Within 100 years, they've managed to push oil content up, you'll see, to nearly 25%. That's massive. This works because the percent of oil content is variable in corn and has a genetic basis. It's genetically based. This is why we geneticists get so worked up about measuring things like heritability, because the genetic basis is really important for the ability to be able to select coral. And so it's select corn, also to select coral. In the simplest way, it works like this. So we see variation in a particular trait in our population. So what I'm showing you here, let's pretend these are all individuals. The x-axis is going to be some trait we care about, and the blue individuals have higher trait content, higher trait values than the gray individuals. Let's say if this was height, the blue would be the tall people in the room, right? We know that offspring traits tend to correlate with parent traits. Tall parents tend to have tall kids. What's cool is that the slope of that relationship, if you were to draw a correlation between the parent trait value and the offspring trait value, the slope of that relationship, or the heritability, is actually the proportion of variation in that trait that is a function of the genetics. When you take that heritability and you multiply it by the selection differential, or basically like how selective you are. So this is saying what's the mean trait value in the population to start with versus the mean trait value, of just those selected individuals. Multiply that by the heritability, and you can actually predict the response to the selection, or how much the trait value should change in that next generation of offspring. So this is incredibly powerful. This is really useful, right? If we know two things, one, how choosy we are, the strength of selection, right? If we have our distribution of individuals, do I take the top 25% of performers, or do I just take the top 10% of performers, right? In that way, I can increase my outcome in terms of selective breeding. The other thing that's going to limit it is that heritability value. How much of the trait is a result of genetics, right? So it's those two things together that gives you the response to selection. So this is the framework that we work under for crop plants, for animal husbandry. Let's think about how we can do this in coral. And as a for example, I'm going to pick one of our favorite traits because we know that temperature tolerance is going to be really important in the response to future climates. So what is the genetic basis of temperature tolerance in coral? One, I want to focus on the genetic side just because the selection side of that equation is fairly easy to do. That's something we can tweak with every cross. But what do we know about the genetic basis of temperature tolerance in coral? Well, most of the media will portray it as this binary trait. Coral either bleaches or it doesn't. And simple categorical traits like that tend to be the result of variation in one, maybe two genes. They're easy to categorize, easy to predict. You did the math on this in your basic biology class. Everybody's having terrible flashbacks right now. But recent research has shown us that's not actually true, right? Bleaching isn't a binary. There's actually tons of variation in the bleaching response, and I just want to highlight a couple recent examples, primarily because the authors are actually here at the conference, so if you want to talk to them about it, you can. This first image is from Adriano Jimenez and colleagues, where they were looking at the variation in temperature tolerance in just a single population of Acropora digitifera in Palau. And you can see this distribution is massive in terms of like highly tolerant to low tolerance individuals. Just here in Florida, Ross Cunning led a very recent study looking at the distribution of temperature tolerance in Acropora cervicornis among the different Florida nurseries and found abundant variation both among and within nursery populations. So bleaching responses are actually a continuum, a continuous distribution of traits. And traits like this tend to be driven by variation in many, many genes. So yeah, you can draw that Punnett square if you want to, but it tends to get really big really fast, right? This silly example is considering variation in just 4 genes with 2 versions of each gene, and already you can't even read the text on that. But in reality, the studies that have gone to look for genetic variation, what is the genetic basis of temperature tolerance in coral, have found that, again and again, it's many, many, many genes that are contributing to this, each gene having a very, very small effect on temperature tolerance. And the challenge there is that, one, just based on genetic information alone, it's going to be really hard to predict temperature tolerance because each of those genes just has a really small effect on the actual trait. And two, it's going to be really hard to engineer a coral because it's going to require you to change hundreds of genes to achieve even some measurable impact on phenotype or the trait. And the other thing that thankfully everyone else has set me up for today is the idea that coral isn't just defined by the host animal. We know that a lot of the variation in temperature tolerance is actually a function of other associates of the coral, the proportion of symbiont types. And I think this result is actually really nicely demonstrated in a very recent paper that was trying to look for the genetic basis of temperature tolerance using some of the most advanced techniques, so whole genome resequencing of coral. And this plot is showing you the proportion of variation in bleaching, the continuous proportion that can be predicted by each of the individual factors in their study. So it's saying, how good can we get at predicting temperature tolerance using each of these combinations of variables? And what you can see is that one of the biggest boosts in predictive power isn't actually host genetics, it's the proportion of symbionts. The proportion of durastinium in the coral is actually a better predictor, or gives you more predictive power. And that doesn't even consider some of the other members of the microbial community, which we just heard about from Nikki, but also which was really nicely demonstrated in another publication by Erica Santoro and colleagues, where they actually showed that you can change bleaching responses just by adding different microbes to a coral. So the host genetics, yeah, influences bleaching, but it's also this other non-genetic component, the symbiont. And that hasn't even considered additional environmental effects, which we also know can influence the response to bleaching. So if you, going back to that plot from Fuller et al., the other biggest boost in predictive power is actually the environment that the coral was sampled from. And this is even reinforced by other studies. I wanted to highlight the work of Flor Drury and Diego Learman, again, a study that was done here in the Upper Florida Keys, where they were trying to say what are genetic and environmental effects on bleaching that we see in sites in the field. So this little plot is showing you each column is a different coral genotype, each row is a site, and it's saying how correlated are those bleaching scores of the different genotypes across site. And if there was a really strong genetic component, what you would see would be really bright vertical stripes, that it wouldn't matter where the genotype was, they would have the same bleaching across sites. And that's not what you see, right? You see a big patchwork, and if anything, the rows are actually kind of brighter. The side effects are actually predominating that bleaching response. So the whole take-home in all of this is that, yeah, there's variation in bleaching that's driven by all of these different factors. But the thing about the selective breeding scheme is, if that variation is not attributable to host genetics, you can be as selective as you want. You can take the top 2 coral in your nursery and you will never change the trait value in the next generation. There has to be a host genetic component in order for selective breeding to work. Whoa, just did something crazy there. Just pushing all the buttons. And the other idea is that, bleaching tolerances that we see in lab conditions might not reflect performance out in a natural environment. So that's something else to consider. And furthermore, the natural environment, as we know very well from all of the talks that have come before, it's not just defined by variation in a single axis. So even if we were to engage in breeding for a specific trait, that might not work when we consider all of the other traits that are exerting, all of the other conditions that are exerting pressure on coral, right? Acidification, pollution, storms, disease. Are there trade-offs among traits that might limit performance? So what I mean here is, okay, say we pick a winner for temperature tolerance. That might not necessarily be the top performer under an acidification scenario or in response to disease. Now, there hasn't been much work on trade-offs to date. I will say, though, that actually the evidence in lab-based studies is positive. So I want to highlight here, I was fortunate enough to collaborate with Dr. Erin Muller. Couldn't be here today. She's riding out the storm up in Sarasota. But what we were looking at was whether you did see evidence of trade-offs. So we put 12 genotypes of Acropora cervicornis into temperature stress, acidification, and then a combined treatment, and asked, like, what's the ranking of those individuals? So even though we saw that the combined stress exerted synergistic effects, synergistic negative effects, so here I'm showing you response to acidification, not so much. Temperature was a bigger drop in the chlorophyll content or the bleaching response, and the combined had an even bigger impact than either of those two things alone. What we actually found was that when you looked at the relative rankings of who was winning in each of those conditions, we actually found that the correlations were positive, or you tended to find that the same genotypes were the winners under those different stress scenarios. So that's encouraging, and similar lack of trade-offs were found in a couple other studies. But again, this is something that we still need to take into field conditions to actually road test it. I wouldn't necessarily make major decisions about this with just an N of 3. The other thing that I've kind of been glossing over is, what's the metric we should even use to measure bleaching tolerance, right? If I'm going to draw a trait correlation between parents and offspring, I better have some idea of what my trait actually is. Should we define bleaching tolerance as the pigmentation score with a color card? Should we do it maybe as the effective quantum yield? What about the symbiote to host cell ratio? And independent of the actual traits, how should I actually be measuring my temperature tolerance? Should I be doing it with acute short-term assays, or should I be doing it with longer duration exposures that more closely mimic natural conditions? We don't really have a consensus idea of what traits we even want to use to define bleaching tolerance, how precisely those can be measured, or even what kinds of assays we should be using that are most reflective of temperature tolerance in coral. So we really, really critically need more data. So for all you young scientists out there, I would love it if you could start working on these questions. We need to know what are the best traits for bleaching, what assays are the most appropriate. And in addition, I've taken a really narrow view with these examples, right? I've been focusing entirely on temperature stress, and that's kind of by necessity because we just don't have a lot of data for other key traits that might be important, like, as Nikki was saying, disease resistance even. So it would be lovely to know, like, what is the genetic basis of other key performance traits? Are there trade-offs? Are there interactions in terms of how different genotypes perform in different environments that's going to affect our ability to really predict outcomes in lab conditions? And so for all these reasons, we as the genetics working group think it's a bit risky to engage in strict selective breeding right now because of all these unknowns, but also because selective breeding, by definition, actually reduces the genetic diversity of your population, right? Because you have this whole distribution of individuals, but you're only taking a select few, right? That little blue shaded area. Those are the only ones that are moving on to the next generation, and you're kind of ignoring the rest of that diversity. And that's something that we've actually seen that can be exacerbated by just doing laboratory breeding. This is summarized really well, I think, in a recent chapter that was written by the Bombs Lab, thinking about managing genetics in restoration breeding programs. So I really encourage you to check it out. But what they explain there is that in natural populations, we know reproduction is really diverse, right? Everybody's releasing gametes. Those gametes have an equal chance of mixing, fertilizing, going on to produce the next generation. And yeah, maybe there's random mortality or some environmental filtering, but if you actually go out and measure relatedness among individuals, generally you find that they're not really closely related in natural populations. Whereas looking at a lab-bred population, there's a lot of bottlenecks in that process just because of logistics, right? You can't possibly collect spawn from every single coral on the reef, so already you've picked who's going to be your subset of parents. You can only rear so many crosses even in your lab because you just don't have that many bowls. And even though we think these conditions are super benign, right, they're still going to possibly select for the larvae that are best at surviving in a little plastic bowl, right? That doesn't necessarily reflect the conditions on the actual reef. And what we see from actual genetic analyses is that when you measure the resulting recruits that come out of that, there can actually be a really high degree of relatedness in those. So even though you think, even if you did a batch cross, you're likely getting very high levels of siblings in your crosses, so you're only selecting a very small bit of that parental pool. And this is a problem because we still don't know what the future is going to hold. So this, I like this panel. It's a recent modeling study by Katie Lauterhouse and colleagues that's sort of asking about what degree of climate novelty can we expect in the future relative to what we've experienced in the past. So this first panel is showing you the change in surface ocean climate between 1800 and 2000. And the heat map bars, blue is a high level of similarity. So for the last 200 years, the change that we've seen in the surface oceans isn't actually all that different. Like, we ain't seen nothing yet. But if you plug this into future IPCC models, you can see that the projected change just this century is huge. We expect that some 83% of climates are going to be novel, or like new, never before experienced, and 95% of existing climate niches are going to be lost. The coral population's ability to adapt to these conditions is a direct proportion to the genetic variation that they currently harbor, right? So if we're limiting that diversity by engaging in kind of selective breeding programs, we're potentially hurting their ability to respond to these conditions that are going to potentially be really different in the future. So maintaining genetic diversity is going to be really essential for the response to novel climate variation. So what do? Right? We all want to do something. How do we maintain diversity while also increasing climate resilience? Because we know that matters. What we're advocating for is not selective breeding, strict selective breeding, but rather managed breeding in natural environments. And the idea here is not us deciding what we should be selecting for, but letting the site of restoration decide who's going to win, right? Because that's what's really going to matter in terms of survival and reproduction. So you've put your corals out at the restoration site. Maybe not every genotype survives or reproduces, but the ones that do are going to be the ones that are fit at that site. If you want to increase temperature tolerance, say, you can engage in things like assisted migration or translocation, where you're bringing individuals from different sites, introducing them, with the idea being that if you're living in a naturally warmer site, you're probably going to have genetic variation for surviving in that site to begin with, right? So that's bringing that genetic variation to a new site, and then whoever lives in those conditions is also going to have the genetic variation to respond to local stressors specific to those sites, right? So it's okay, like not everybody's going to survive, but whoever is surviving is going to be the ones that are going to be doing the best. And so when they reproduce, they're going to have offspring that are going to be adapted to the local conditions, potentially also bringing along these temperature tolerance alleles, and then continued rounds of introduction and breeding is going to maintain that diversity while potentially also boosting tolerance. Now we realize that translocations are risky, and so we're also really strongly advocating for a comprehensive and clear genetic management plan to guide these efforts. But I do want to point out that translocations aren't this nutty idea that are only being considered for coral. A really nice review by Ben Novak and colleagues actually looked to say, how often do we use translocation in the management of endangered species? And it might be more than you think. So for 30% of taxa that were previously listed as endangered species and have now been delisted, we use translocations in 30% of those. And of remaining listed taxa on the endangered species list, we are at least considering translocations for another 70%. So these are interventions that are being actively used in other conservation management approaches. And we think they should be considered for corals, especially in areas where we know that diversity is limited, where we've seen big demographic declines, like, for example, acroporids here in Florida. We really need to make sure that we're accounting for relatedness, particularly in the sexual breeding programs that different organizations have been engaging in, and population simulations can help manage some of these risks given certain demographic information. But I want to emphasize that this isn't simple. This isn't a simple side project undertaking. Like this is going to need a reasonable investment of time and money to support someone actually doing these plans. And we're actually really encouraged by the work of Kate Rodriguez-Clark, who's going to be speaking next. She's sort of doing the preamble to a plan, but we really hope that that can continue to be supported because we see this as a really essential part for the management of coral species going forward. So with that, I just want to close by reminding everyone that we do have another publication that we put out. Ileana presented this at the 2018 ReFutures Conference, and I wanted to raise it again because everybody keeps talking about that magic number of coral that you're supposed to have. This is the paper if you want to look at those recommendations. If you're wondering, the magic number is actually about 5. And I don't mean 5 genotypes only. What I mean is that, given your set of reefs at your site, what we would love is if you could start your nursery with 5 coral from each of the different reefs in your zone. So that would be like the really lovely reef, but also the really hot reef, and maybe the reef that also has a lot of sedimentation from the local dredging, right? Take 5 from each of those, capture that diversity, and use that to start your restoration population. But there's lots of other recommendations in here if you'd like to see that. And so with that, I thank you for your time, and I'd love to take any questions.
Thank you so much, Carly. That was great. I definitely learned a lot.
Great.
And we already have a question from Facebook, so I'm going to go ahead and ask it. It's from Sage Morningstar. She asked, what could, I'll add, possibly be the downsides of translocations? Do we have any examples of detrimental translocation events in conservation?
I would say yes, certainly. So when we say translocations, I'm not talking about like moving different species in, but it would be, possibly taking Acropora cervicornis, like I said, from Cuba, moving it to Florida. Some of the downsides could potentially be bringing other unintended partners along with that, right? Say we know disease is prevalent right now and you're unintentionally maybe spreading disease if you're bringing individuals. So you do have to do some checks to say, is that going to have a negative impact? In terms of genetic impacts, given how sort of well-mixed corals are genetically because of that broadcast spawning, we're not too worried about the genetic impacts, even on a Caribbean-wide scale. Mostly it would be these other potential invasives, like maybe spreading Drupella or some other disease, and so I think there would have to be a careful quarantine plan.
That makes sense. People are welcome to ask questions in the room. Ilsa said, fantastic job, Carly. Go ahead.
Hi, Carly. That was a great talk. With the translocation, what would you recommend as some of the health metrics or traits, typical traits, other traits that we can measure that would be useful to measure, after a translocation process? So I'm guessing you don't just want photomosaics, but if you're actually trying to account for all these potential health changes and artifacts, what would you recommend?
You mean in terms of once the coral have been brought to the site, how are they doing?
Yeah, exactly.
Okay, so we in the 2019 publication, we if you have time, we advocated for things like if you could get some estimate of growth, maybe disease resistance, fecundity once they've gotten to the spawning stage, and that could be something like okay, they're spawning, but maybe how many gametes are they producing? So just some traits that we think are fairly central for coral performance. But again, I don't think it's a necessity, actually. It's more, if you know who your genotypes are, which I hope everybody is tracking who their genotypes are, it's more knowing that like, okay, genotype 5 is still alive, genotype 7 is still alive, genotype 8, right? So they'll probably reproduce. Whereas if you're in a scenario of like, okay, well, we only have genotype 8 left at this site, there's not going to be any sexual reproduction happening there. So in that case, you would need to bring in some more genotypes to say, okay, well, 8 was the only good performer that batch, so maybe I better go get like 62 and 54 and try them out next.
Great, thanks. And just a follow-up too, I love the transplant from Cuba to Florida, but wow, that sounds pretty politically tricky. So how well are we set up for even this kind of maneuver? I mean, obviously, maybe between different sites in Florida, but can you just comment on what's needed politically to make something like that happen?
No, you raise a really important point, and I think Andrew Baker can probably speak more to this. He's been struggling with this a lot for his Ruth Gates project. But yeah, it's going to require international collaboration, and that's something where I think in the Caribbean we have a much bigger challenge than, say, folks that are managing the Great Barrier Reef, right? Because they're just dealing with one political entity. So I think there is going to have to be some partnership or maybe even some reciprocal exchange considered so that everybody feels like they're getting equal access to genetic resources that might be really important in the future. And I'm not a politician, like I'm a scientist, but I sincerely hope that there are folks here who interface more on the political side of things that can help facilitate those conversations that need to be had.
Thank you.
Go ahead.
If you're willing to speculate, how many other heritable traits do you think follow a similar pattern with temperature tolerance, in which there are many, many genes and they're only contributing in a small way? And do you think that the marine environment and the hull body structure are perhaps contributing to the evolution of the genes in such a way?
I would say, for the second part of your question, yeah, I certainly think that host and symbiont are adapting and evolving together. And so that's another layer of selection apart from the abiotic environment, like the temperature, acidification, everything else. In terms of the genetic basis of traits, I think it would be, for those traits that we see that are either categorical or continuous, that can be kind of a clue. So I'm not a disease researcher, but if it's something that's more along the lines of like, okay, well, the coral either gets the disease when I try to infect it or it doesn't, that could be a much simpler control, at least on the host genetic level, if that's the case. But again, that's why we need more of these studies to try to say, like, well, one, how much variation is there even in this particular trait? Is it more than what we thought initially? And studies to try to ask, what is the heritability? And yeah, thanks there. Thanks.
Thanks for your questions. I have a few questions of my own, and I can comment on the other, the political piece that someone just asked a question about, just because I spoke with Andrew about this yesterday, and that it's possible that it might not be quite as complicated as we thought, and possibly through CITES and cryopreserved material, we can actually make these exchanges without too many politics. So Jen Moore and Andrew Baker are people to follow up with about that, but don't be too scared off. I was going to ask you, Carly, about your how much is what you said about creating these genetic management plans really specific to the Caribbean versus what we heard from other basins where they really do have, corals that are, relatively healthy, they have higher population numbers, higher species numbers, all that kind of stuff.
I think it very much is specific to the Caribbean, like sort of when we're in the point of like the demographics have declined so much, right? When we're doing things like saying, let's collect all of the remaining genotypes of Palmata in Florida, right? Like that's at the point that we're like, okay, now we're really managing these. And in that kind of a situation, yeah, we've really got to think about how much genetic diversity is left and being careful not to lose that. Whereas the beautiful images of Indonesia, we can go out and like try to assess genetic diversity, but I don't think there's any need to manage it yet, because one, there's probably even more diversity than we've even measured with cryptic species and other things there. So yeah, I think we're at a different stage here in the Caribbean for sure.
Yes, go ahead.
Carly, thank you so much for that excellent talk. So I work in Broward County at Nova Scotia University and run one of the land-based nurseries we have. So I get asked multiple times if I do selective breeding. I always say, well, the corals from Broward are already super corals because they've gone already through so many stressors. So actually, the corals that I have, whenever they spawn, I put everything together and raise the larvae of all this mixing. So my question is, though, I am obviously, as you mentioned, reproducing the same corals every year. And as Ileana has done a study about how many genotypes we should have from each site, I also wonder if there's any study already about how often should we change our rootstock so that we are not making one genotype more prevalent in the population of what we're putting out there. So I guess we need probably somebody that also looks into that externally so that we have when should we change our rootstock, when should we change some corals in between different land-based nurseries so that we can guarantee that the roots we're putting out there, we're not also making some genotypes more prevalent than it should be.
Yeah, so I think me personally, Kate might have a different answer to this in her next talk. But I would say it's something that probably as frequently as possible. And actually there's some things you could probably do now to try to say like how genetically related are these offspring. So one easy test that doesn't even involve sequencing would be something like individual fertilization trials, right? So in your set of parents to say like who is actually able to fertilize who, because that would tell you like, oh geez, actually like only this subset of individuals is actually compatible and they're not even fertilizing with these other ones. So that would give you some idea of like how diverse possibly are the offspring. You could also try to do things to increase the diversity, like instead of doing a bulk cross and like hoping everybody's fertilizing with everybody, it's a little more painful, but like do all the individual crosses and then pool them together, right? And that way you're at least saying, okay, everybody's fertilized with everybody else and maybe that's more diverse, but yeah, I would encourage like reshuffling the deck as often as possible.
Yes, I guess it would be nice to have some estimate of how often we should do that and how big should be our original rootstock, and I guess some sort of open genetic space to inform what we can do. Thank
Yes.
you so much, Carla.
Genetic management plan. Yeah.
Exactly.
Thank you.